Okay, today we are going to fill out pages 16, 17, and 18 with 2 screencasts and an Amoeba Sisters. So let's get started. Number 1, DNA holds the code. We can also call that OR recipe if you want something a little bit easier to understand. For all of your human proteins, there's about 21-ish thousand human proteins. And if I had to describe the 2 things that they do in your body, I would make 2 categories. They control your appearance, skin color, eye color, many more things like height, and your health. They are your body's enzymes, hormones, and antibodies, etc. Also your hemoglobin, so for sure need proteins. We'll get more into that on page 17. All cells have an entire copy of your DNA. So if you go down here to just a generic cell, you look right here into the cell's nucleus. And in each human cell in the nucleus, you have these chromosomes. You can zoom in right here to these karyotypes, which we talked about first semester. You have 23 pairs, and each pair has 1 copy from dad and 1 copy from mom. Okay, so a little bit of context here. Every single gene varies in what ATCGs make it up. So let's go up here. The average human protein coding gene is about 3,000 letters long. What do we mean by letters? Well, A binds with T, adenine binds with thymine, and cytosine binds with guanine. Those are the 4 options. Okay, so the shortest gene in your body has about 500 letters long. So this is an example of the ATCG sequence for a random gene. And the longest gene is 2.3 million ATCGs long. So this is just for context. Another piece of context, of all of your DNA in your body cells, less than 2% of it actually codes for a protein. The other 98 up to 99 is called junk DNA. Okay. Every person has 2 copies of each gene, 1 inherited from each parent. Okay. Cells are different from each other based on the genes that are turned on. Okay, so cells are different in 3 ways. Let me show you. The first way that they're different is what they do. You guys can see here, a red blood cell does something different than a brain cell. They have different functions and obviously very different shapes. So that's the 2 differences right here. The shape and the function. The next difference and final difference is which genes are turned on. What do I mean by that? Here is one of the thousands of genes that you have. Okay, remember all these little lines on the chromosomes? Those are all genes, okay? So every gene has a promoter, which we'll call the on switch, the coding region, which is the true recipe, and the termination sequence, which is the off switch. Okay? So let's see, let's go down here. Let's zoom in on, if we could, one little line on one little piece of genetic material, this little chromatid. This one's probably from the maternal egg, this one's from the sperm. Zoom in on one little part of it. Zoom in on here. You're looking at one gene. This is what we call a genotype. A genotype is a gene's sequence of adenine, thymine, cytosine, and guanine. Okay? And after we finish learning this unit, we'll discover, which I'm pretty excited about, how these letters, how that code is translated into what these are right here. These are just very few examples of something called phenotypes. Phenotype is a physical expression of a genotype. And these are kind of fun to see if you have them. So this is the physical expression of a genotype. Okay, so here are some examples of some phenotypes that were coded for by your genotypes. Do you have a widow's peak, or do you have a straight hairline? Can you roll your tongue or not? When you cross your fingers, what thumb naturally goes over the other one? Do you have a bigger second toe than a thumper? Do you have a bent peaky? Do you have this little point right there in your ears called the Darwin point? Are your earlobes attached or detached? Do you have a space here? What's really cool, what I love about this unit is like, how in the world can this sequence of ATCGs, how can that produce this? So the first piece of that is called transcription. So we'll finish this page with number 5. DNA is like a recipe book, and mRNA is an individual recipe that tells cells how to make a protein. Proteins produce traits such as eye color and skin color, etc. Transcription, you may have remembered this from a previous science course, is the process that produces a disposable copy of a gene. Okay, so transcription goes in, finds a gene that you want to code for, and creates like a disposable, very cheap actually, copy of a gene to be sent to the cytoplasm ribosomes to become a protein. Okay, this little disposable gene is called mRNA because it sent the messenger between the DNA in your nucleus to the machinery, which is like the ribosomes. So this is the messenger. DNA is kind of snobby. It doesn't like to communicate directly with the ribosomes. It has to send a messenger. Okay. Oh, I got this mixed up. The recipe is called mRNA. Lowercase m, capital R, capital N, capital A. We'll talk about RNA in a minute, where M stands for messenger. Okay. So that's just the basics of how you have DNA, and each gene is made up of ATCGs, different amounts, different sequences, and those genotypes are going to cause these physical manifestations called phenotypes. Turn to page 17.